You should be able to see the test, can you?
Yes, we can see it.
Okay. All right, the average for this test was 72%. The highest grade was 95. So let's take a look at the various questions and answers. The first one is about the genetic code, and it's something that you really all ought to know. About half of you got this completely right. A few of you got the transcription and not the translation, and some of you didn't get it at all. So just to remind you, A's and T's, G's and C's. T always specifies A and vice versa. G always specifies C and vice versa. So the DNA sequence given here, TAC, the first codon, T specifies in RNA an A specifies a T. But remember, RNA does not have T's, RNA has U's. So every place in this DNA sequence that you see an A, in RNA, that would be a U. So the first codon, the C, specifies a G. So the first codon in RNA would be AUG. The second one, AGT, would be UCA, and so on. I'm not going to go through the whole thing. But then to translate this, some of you did not apparently know how to use a codon table. And this is important. So a codon table has a first base, a second base, and a third base, referring to a particular codon. So if you take the first codon in RNA, AUG, so the first base is A. You go down to A, and the second base is U, so you go to the first box to the right, AU, and the third base is G. Third base is all the way over to the right, third base is G, so you go back to AUG, that's methionine. And similarly, if you go to the second codon, UCA, and go to the first base, U, it's up here on the upper left. The second base is C, you move to the right. And the third base is A, here. In this box, they all specify serine. And that continues. First base, second base, third base. So the protein or the peptide sequence you should have come up with is methionine, serine, asparagine, cysteine, arginine, and a stop codon. Are there any questions about this, about how you do it? It may come up again, so please make sure that you do know how to use a codon table and how the genetic code works. Okay, the second question is about protein structure. And it says, the blank structure of a protein refers to specific folding and bending of the primary and secondary structures. So that immediately tells you that the answer is not primary or secondary. And because of the folding and bending, it is tertiary. In the second blank, some proteins have a structure that occurs when two or more peptides come together. Well, anytime you have two peptides, it's not going to be primary, secondary, or tertiary, so that leaves us with quaternary. The third question, I wanted to get a perspective on DNA, the copying of DNA. And remember, we went through all the enzymes involved in the production of new DNA fragments. And so part of that is has to do with the Okazaki fragments, and that's what this question is about. So what is the sequence of steps dealing with Okazaki fragments? Well, it's in red here. First, you remove the RNA primer, then synthesize new DNA, and then DNA ligase comes along, stitches together the fragments to make a complete DNA sequence. Any question about that? Then we had a question about coronavirus. Most of you got this right. The virus particle has on its surface a protein called spike, which interacts with a protein on the surface of human cells called ACE2. And the answer is really given following the blank, angiotensin converting enzyme, ACE2. Then a question about cystic fibrosis. The CFTR protein transports chloride ions. Then the next question is about the digestive system, which we went over in some detail, having to do with cystic fibrosis. And I ask you to match up the features on the left with items from the list on the right. So exocrine cells produce digestive enzymes. Zymogens are inactive forms of enzymes. Bile acid, remember, the acid solubilizes fats. Endocrine cells are the ones that make hormones, and the gallbladder produces bile. Any question about those answers? The next question is about a karyogram, or karyotype, and it's false. It states the karyogram is a scanning electron micrograph of an organism's chromosomes. In fact, you wouldn't use a scanning electron microscope to look at a karyogram. You'd use a compound light microscope. And any time that a scanning electron microscope would be used as a secondary step, in other words, you take a look with a light microscope, you might want to have a closer look with a scanning scope, but that would not be a typical karyogram. Then the next question is about the bonds, chemical bonds found in the tertiary protein structure. Most of you got most of this right. The four bonds I was looking for: ionic, hydrophobic interactions, hydrogen bonds, and disulfide bridges. Of these, disulfide bridges are the strongest because they are covalent. Now, some of you indicated dispersion forces, which is another name for van der Waals forces. And I accepted that because even though these are very weak bonds, they are sometimes involved in protein tertiary structure. So if you said dispersion forces instead of ionic, I accepted that. Now the next one is about mitosis. This is mitosis, not meiosis. It's another matching question. What is anaphase? The chromatids move toward the poles. Interphase, DNA replicates. Telophase, the daughter chromosomes arrive at the poles. Metaphase, the chromosomes align in a plane at the center of the cell. In prophase, chromatin condenses into chromosomes. The next question, I think everybody got it right. It's true, mitosis is asexual, meiosis is sexual. The next one, there was some confusion. There are two phases in meiosis. in which genetic variation occurs. They are prophase and metaphase. Now, some of you indicated recombination and reassortment or random assortment of chromosomes, which are not wrong answers if it were the right question. But here the question asks about phases in meiosis, and those would be prophase and metaphase. Then why is meiosis important in evolution? The essence of the answer here is meiosis generates variation at every generation. And the second part that we talked a lot about in lecture is that natural selection then uses the raw material that's generated by meiosis to sort out the fittest individuals at any given point in evolutionary time. So I gave, so some of you got full credit on this. I gave partial credit for partial answers, but these 2 points is what I was looking for. There are several sources of genetic variation, and I wanted you to pick those out. They are mutation, chromosomal recombination, and random chromosome assortment. Most of you got that. And then we have the question about 5' and 3' ends. Again, I've asked you this before. This turns out to be false. The 5' end is a phosphate group. The 3' end is a hydroxyl group. It's not glycosylated. So that's false. Now, when comparing meiosis 1 and 2, there are differences and similarities, and I ask for the statements that are correct. The chromosomes split, and chromosomes assort randomly. Now, the next question is about membranes. We have 4 major classes of biological molecules. Which one of the 4 is not found in membranes? It's nucleic acid. And then I told you I was going to keep asking about RNA until you all get it right. And here it is. So which one specifies amino acids? Transfer RNA. Machine that builds proteins, ribosomal RNA. The code for the protein sequence is messenger RNA. And which one has catalytic capability? It's ribozymes. Most of you got most of that right. So I can stop asking it now. Then there was one about osmosis, 2 beakers joined by a tube with a semi-permeable membrane, permeable only to water. Left beaker has 50 mils of 10% sucrose. The right beaker has 1% glucose. You leave it overnight. What would happen, and the answer is the level of liquid in the left beaker would go up and the right beaker would go down due to osmosis. Active transport, next question, number 19, moves ions across a membrane against a concentration gradient, but because ions carry electrical charges, there's usually an unequal distribution, which imposes a second gradient, which is electrical. And the combination of these 2 is called an electrochemical gradient. And the next question is also about membranes. It's another matching. Phospholipids, what do? They form a bilayer. Transmembrane proteins facilitate passage of small molecules. Cholesterol is a buffer buffers membrane fluidity against temperature changes. The peripheral membrane proteins, their attachment to the membrane is reversible, and the glycocalyx is the sticky coating of protein and sugar polymers. And finally, question about CRISPR. I think everyone in the class got this right. It's true. The statement about CRISPR with a guide RNA and an enzyme that makes a double strand cut, that's a true statement. And okay, so I want to come back to CRISPR in a minute, but before, is there anything about the test that you have questions or comments about? Okay, if not, I'm going to stop sharing this screen. Okay, do you all see Nobel Prize in Chemistry?
Yes.
Okay. Now, those of you who were paying attention to the news last week may have noticed what's on this slide. The 2020 Nobel Prize in Chemistry is for CRISPR, which we talked about in some detail in an earlier class. So this woman, Jennifer Doudna from Berkeley, shared the Nobel Prize in Chemistry with a French woman named Emmanuelle Carpentier. So you just see pictures of them, of Jennifer Doudna in her lab and holding a space-filling model of a CRISPR-Cas9 complex. And just to remind you, the beauty of CRISPR is, first of all, its simplicity. Secondly, its specificity for targets. And so there are really just two components to CRISPR, a guide RNA, which you make in the lab, and you make it to correspond to some point in the genome that you'd like to mutate. So it will bind to that point, and then this enzyme, Cas9, comes along and makes a double-stranded cut in the DNA that you've targeted. And that cut then offers a lot of opportunities. It can be left and allowed to fill in, which would probably lead to mutations, or you can put a piece of DNA of your choosing at that point. So this specificity is one of the reasons that CRISPR is so important. The other is that it's universally applicable. That is, it works in, as far as I know, every organism that it's ever been tried, including prokaryotes and a wide range of eukaryotes. It's already been used to improve crop plants. There's even speculation, there's a group at Stanford now that is trying to develop CRISPR as a treatment for coronavirus. Okay, that's just an aside I wanted you to know before going on to the topic of the day, which is Mendelian genetics. I want to put this in a historical context. So in the mid-1800s, there were some great ideas that are still affecting us today. So I put on the right-hand side here, Karl Marx was a great thinker of that time. He published the Communist Manifesto, which is still influencing the political systems of a significant part of the world's population. The American Civil War occurred during this period, another major event in world history. And it's during this period that Gregor Mendel and Charles Darwin were doing their seminal work. Gregor Mendel was an Austrian monk who lived in an abbey, and he published his work between 1856 and 1863. Now, of course, at that time, he had no knowledge of DNA or genes or chromosomes. He used simple garden peas to determine the laws of genetics. Now, Charles Darwin was an English naturalist living at about the same time, but the two of them, as far as we know, did not have any knowledge of each other. Darwin published his Origin of Species, which introduced the theory of evolution, in 1859, and coupled to that is this concept of natural selection, which we've talked about numbers of times in terms of meiosis. Now, Mendel, even though he published in the mid-1800s, no one paid any attention to his publications for about 40 years. And then around 1900, there were three scientists independently, Dutch, German, and Austrian, independently rediscovered the publications. They tried to repeat Mendel's results, and they were successful. They confirmed what Mendel had reported. So why do I bring these up together? Because the relationship is here at the bottom of the slide in yellow. Darwin established natural selection as the force controlling speciation, and Mendel provided the rules for it, the basis for variation that underlie natural selection. So the two go hand in hand, even though the individuals who came up with these great ideas didn't work together, didn't know each other. Now, prior to Mendel, what was the nature of genetic analysis? Well, genetic analysis has really been done throughout agriculture for approximately 13,000 years. And when humans specifically took organisms like plants and crossed them with each other, they would get something like what you see here. Cross two parents with different traits. In the progeny, there is a continuous variation of the trait. In other words, no segregation into distinct groups. If you look at this bell-shaped curve, at the progeny of such a cross, there'd be a few individuals in the progeny that would be mostly like one parent, and a few mostly like the other parent, but most of them would be in between. They would look like a mixture of parental types. So that doesn't really tell you much. It just tells you that you've mixed up the traits of two parents and nothing more. So that's prior to Mendel. What did he do that made an advance? Two things. One is inbreeding, and the other is analysis of F2. Now, these don't mean much to you now, but as we proceed, they should. So first, some definitions. What is a cross? It's the breeding of two plants or animals, parent 1 and parent 2. The parental generation is called the F1. The original two plants are crossed. So the F1 generation is the offspring of the first cross. F stands for filial, the Latin word for son. Then if you take two individuals from the F1 generation and cross them with each other, you get the F2. That was a critical step for Mendel. Two more definitions, phenotype and genotype. Phenotype is what you see. It's the characteristics of the organism that are seen or produced by the genetics. Phenotype is always descriptive words. Genotype, on the other hand, is a symbol. It's a representation of the actual genetic makeup of the organism, represented by letter symbols. So these would be words describing phenotype and symbols describing genotype. So then how did Mendel do his experiments? Well, the key step one was, before he started doing his crosses, he developed true breeding inbreds. What is an inbred? It's a mating of closely related individuals so that after several inbreeding generations, the genetic heterozygosity is gone. We'll talk about heterozygosity in a little bit. So all the loci are homozygous, and the result is a so-called true breeding line. Well, what is a hybrid? It's a mating of distantly related individuals so that most of the loci are heterozygous. When two inbreds are crossed, it can lead to a condition known as hybrid vigor. Now, essential to Mendel's success, he inbred each of 7 P characteristics, so that now all the loci for all 7 of those characteristics were homozygous, no allele variation. And it's only after he had laboriously done this did he start making crosses between individual Ps having different traits. Now, on the right here is a diagram of a pea plant, the pea flower. Peas are self-pollinators. So they produce both pollen and stigmas within the same flower, and fertilization occurs before the flower even opens. And that was an issue that Mendel had to deal with. So I'm going to talk a bit more about inbreeding, which is one of the keys to Mendel's success. Without it, his distinct segregation patterns would not have been seen. So just to reiterate, inbreeding is a mating of closely related individuals. For example, in humans, it could be cousins, or brother and sister, or self-fertilized plants. It's mating which increases the number of individuals that are homozygous for a trait, and therefore increases the appearance of recessive traits. So what is an inbred then? It's an individual that's resulted from repeated inbreeding. And usually to get a true inbred line, it takes at least 6 generations of inbreeding, so that the plant, the inbred is homozygous at most or all loci. Okay, any question about what inbreeding or an inbred is? Now, I'll make a short digression from Mendel and talk a bit more about inbreeding and hybrid vigor. Inbreeding often leads to a condition known as inbreeding depression. And that is a loss of vigor or function as a result of inbreeding. So inbreeding is not necessarily benign. So here I've tried to illustrate what inbreeding depression and hybrid vigor are. So on the left, you see a plant from a natural population, and its chromosomes are represented at the bottom. So there are blue sections and white sections in both chromosomes. So these would then be called heterozygous chromosomes. Now, if you inbreed, in other words, you take two plants from the natural population, cross them, and then take their progeny and cross, take their progeny and cross, for a number of generations, that is inbreeding. And what you normally get, especially in a plant like corn, which this is inbreeding depression. So now, because you've inbred one inbred plant after a series of breeding steps, has all white chromosomes, so they are homozygous. The other inbred plant has all blue chromosomes, so they are homozygous. But you notice the plants are small. That is the effect of inbreeding depression. But if you cross these two, that's hybridization, and so the progeny have homologous chromosomes, one of which is all white and the other which is all blue. So they're heterozygous at all loci. And that can lead to hybrid vigor, which is a bigger, more robust plant than the original or than either of the inbreeding depressed plants.
Professor,
Yes?
what is the loci?
Loci, a locus is any spot on a chromosome. Usually a gene, but not necessarily. It just indicates a place on a chromosome. And we'll come a little later and talk more about loci. Now, a point to be made here on the left is that if you cross any two inbreds, you don't necessarily get hybrid vigor. In fact, hybrid vigor of the type that the geneticists are looking for usually does not occur. You have to sort through and identify the particular crosses that will give hybrid vigor. Okay, any other questions about inbreeding depression or homozygosity and heterozygosity? We'll come back to those terms again. Now, here's an example of a major commercial success as a result of hybrid vigor in corn, maize. Essentially, all the corn, in fact, all the corn that we or animals eat is hybrid. So here you see it illustrated. The plants to the far left are inbred, suffering from inbred depression. Plants on the right, same, it's a second inbred, suffering from inbred depression. When those two parents are crossed, you get what you see in the middle, this very big, vigorous plant. And if you look at the commercial benefit of that, and the ears of corn to the right, the two parents make small ears. The hybrid makes a huge ear with a lot more kernels than either of the parents. So that's probably one of the best known examples of hybrid vigor used in agriculture. Interestingly, Darwin, yeah?
That's other known as heterosis.
Yes, hybrid vigor is known as heterosis. That's right. Good point. Hybrid vigor was noticed by Darwin first published in 1876. So hybrid vigor has been known for a long time, and we still don't know the molecular basis for it. But interestingly, Darwin had this comment about it: hybrids such as a mule have greater vigor. And what he said about it is, the mule always appears to me a most surprising animal. The offspring of a horse and an ass should possess more reason, memory, obstinacy, social affection, powers of muscular endurance, and length of life than either of its parents. Seems to indicate that art has outdone nature. And it's true. A mule, for getting work done, a mule is better than either a horse or a donkey, for the reasons that Darwin indicated. Inbreeding affects human populations. Here is an example: the Habsburg jaw. So on the upper left, Holy Roman Empire, Charles V, suffered from an enlarged jaw, known as the Habsburg jaw. Now, I've chosen a bunch of pictures of people from that royal family, his heirs, several generations. They all had one form or another of the Habsburg jaw. And in fact, you can even see what resembles a Habsburg jaw in the population today, as you see in the bottom center. Now, this is interesting. The powerful Habsburg dynasty ruled Spain for nearly 200 years. It ended in 1700 with the death of King Charles II, who left no heirs. The termination of that lineage likely resulted from inbreeding, which probably left Charles II sickly and infertile. And I ran across this. I didn't know that ligers existed, but they do. They don't occur in nature, but in zoos, you can cross a lion and a tiger. And if the male is a lion, it's known as a liger, which is larger than either parent. Interestingly, if the male is a tiger, it's known as a tigon, and it's smaller than either parent, for reasons that no one knows as far as I know. So that was a digression and aside that I thought would be of interest. I'd like to get back now to Mendel and his pioneering experiments. So Mendel chose 7 pairs of characteristics, and he highly inbred all of them to give definitive F1 traits. And they're shown here, listed on the left and shown in his pictures on the right. Seed shape was 1, round or wrinkled. And if you cross round times wrinkled, you got all round in the F1. Seed color, yellow and green. Cross them, they're all yellow. Flower color, purple and white. F1s are all purple. And so on. Pod shape, pod color, flower position, and height. So it was very clear after he had done his inbreeding that he could get a clear-cut result in the progeny of the first cross. So then how did he do crosses? Remember, this is a self-pollinating plant. It actually fertilizes itself before the flowers open. So what Mendel did was open the flowers before pollination occurred, take out the anthers so that the ovary could then be fertilized at a later point with pollen from a different plant. So this shows the use of forceps and paintbrushes, which indicates how tedious this procedure was. Open the flower buds, remove the stamens, and then later dust pollen from one plant onto the stigma of another And Mendel did literally thousands of these. So it took a lot of patience to do his experiments. Mendel's first experiment is called a monohybrid cross. Now, these are phenotypes. He crossed two true breeding parents, one purple with purple flowers, one with white flowers. And in the F1, he saw that all the plants had purple flowers. Now, the key thing that Mendel did next, instead of stopping here, he mated F1 individuals with each other to get the F2 generation. And when he did, he found that the white flowers reappeared. And the ratio of purple flowers to white flowers was 3:1. And so next, so that was phenotypes. Next, let's look at the genotypes. So in this case, the gene is indicated by the symbol big P for a dominant allele, little p for a recessive allele. What are alleles? They're alternate forms of the same gene found at the same chromosomal locus. Alternate forms. So in the parental cross, because they were inbreds, he was crossing a big P, purple, with little p, white. And the gametes from big P are all big P. Gametes from little p are all little p. But now if you cross those, big P and little p, what you get are the F2s, which are now segregating in a 1:2:1 ratio at the level of genotype. Remember, the phenotype is what you see. That's 3:1, 3 purple to 1 white. The genotype is 1 big P, 2 big P, and 1 little p, which is the only one that shows the recessive characteristic. So is that clear to everyone why you end up with these ratios? Okay. So to elaborate on alleles, and someone asked about loci. A locus is just a place on the chromosome, usually designates a gene. So an allele would be a form of a gene on one chromosome differing from the same gene on the other chromosome. So the chromosomes are homologous. That is, they're bred to be the same at all genetic loci, except at this one locus, they're heterozygous for purple flowers versus white flowers. Dominant versus recessive. In Mendel's experiment, the characteristic that appeared as the phenotype in the F1 was called the dominant characteristic. Whereas the characteristic that disappeared in the F1 but showed up again in the F2 was termed recessive. So those you can consider definitions of dominant and recessive, whether it shows up in the F1 or not, and whether it appears in the F2. So this just illustrates what we've been saying. On homologous chromosomes, any given locus can be homozygous or heterozygous, dominant or recessive. So here you have two homologous chromosomes, a pink one and a blue one. At the P locus, it's homozygous dominant. both alleles on both of these chromosomes are big P. At the A locus, it's homozygous recessive. Both alleles are little a. Whereas at the B locus, the alleles are heterozygous. One is big B, dominant allele. The other is little b, a recessive allele. So this hopefully clarifies what loci are, what alleles are, and what it means to be homologous chromosomes.
And I'm sorry, Professor. So did you mention the standard notation for a capital letter to be the dominant and a lowercase letter to be the recessive?
No, I didn't. Why don't you elaborate on that?
Okay. Well, there's really not much more to be said. There is standard notation or some basic rules that get followed. First off, when you're assigning codes, you can assign any letter code that you choose for a particular trait. So you could use, if you have, say, tall and short that you're looking for, you could use a capital T as tall and a lowercase t as short, provided that short is the recessive gene. But my point was that the standard notation is if you see a capital letter, you are talking about a dominant trait. If you see a lowercase letter, you're talking about a recessive trait. So there is some standardization there just for your own edification. Make sure when you're using these, you can decipher them correctly. Easy to show you this. I can cover it in workshop at any point if any of you want to jump in for that. Sorry for the distraction, sir.
Oh, thank you, Scott. Yeah, I thank you for the clarification. That is a very important point. The difference between dominant and lowercase letters is gene symbols.
Professor, what is a homologous chromosome, though?
Homologous chromosomes are homozygous at all loci, with the exception of you can have particular loci at which you have different alleles. So a homologous chromosome would be the result, homologous chromosomes, plural, would be the result of inbreeding, where you take, go generation after generation of selfing, crossing each generation within the population, within the progeny. If you do enough of those, the chromosomes become homologous at all loci. At the level of DNA would mean that the DNA sequence is the same in both chromosomes.
And that's on a per chromosome basis. So chromosome 1 can be homologous with its pair chromosome 1. Remember, these chromosomes come in pairs, and the DNA needs to be the same basic information on each chromosome. So when you are dealing with, say, chromosome 1, you're going to have, and in this case, they've colored it, and if you'll forgive the old-fashionedness, they've got pink. You could say that's the mom's chromosome or the mom's genetic information. The blue is maybe the dad's genetic information. They came from two different sources, but they contain the same basic information at the same basic points. And that's how we get the ability to reproduce. So you could have a homologous chromosome on pair 1. It will not be the same as chromosome 7, say, where the CFTR is found. That would be a different chromosome. However, chromosome 7 could have a homologous, it will have a homologous pair as well, because they have to match up, at least in the basic information, the locations of these genes. Otherwise, we don't get really any reproduction. You can't mismatch chromosomes.
Right. Okay, any other questions about that?
And then
Yeah,
homozygous would mean that they have identical pairs of genes for any hereditary characteristic.
you said homozygous? Yes.
Yes.
So if you're homozygous at a locus, it means you have either two dominant alleles there or two recessive alleles.
Okay.
And it would be, the proper notation would be to say something is homozygous dominant or homozygous recessive. You would not use homozygous by itself unless, it would be unusual. I would, if you were defining things, I would always include whether something is homozygous dominant or homozygous recessive. The only difference with heterozygous is it automatically means that there's one of each. So there is no heterozygous dominant. There is no heterozygous recessive. And you can just use that term by itself. It just means one of each allele is present. But with homozygous, you must, for clarity, include whether you're talking dominant or recessive.
Anything else about this issue? Homologous, dominance, recessive, heterozygous, homozygous, loci. All terms that you need to know and remember how to use them.
Homologous one more time.
Homologous one more time. It means if chromosomes are homologous, they have the same DNA sequence throughout, with the exception that a particular locus can be either homozygous or heterozygous. But homologous chromosomes are the result of inbreeding to the point that the DNA sequence is the same for both.
And that's what this representation shows.
Yes, yeah. The whole point of this slide is to make that point, that these chromosomes, even though one is pink and one is blue, they're the same, they're homologous. It's just that one is from one parent and one is from the other parent. Then on these homologous chromosomes, there are loci that are either the same on both chromosomes or different. And if they're different, it's known as different alleles. If they're the same, if they're homozygous, it means that the same allele, exactly the same DNA sequence, is present on both loci. Okay, genotype versus phenotype. We've mentioned this before, but the phenotype is what you see. And so in this case, what you see is 3 purple flowers and 1 white one, a ratio of 3 to 1. The genotype is a symbol for the genetic condition. In this case, the genotype is a ratio of 1 to 2 to 1. One symbol is P, so it's 1 big P, 2 big P, and 1 little p. And it's only the little p, the recessive, when the recessive trait is homozygous, that you see the recessive phenotype, white. All right, this one is for you to work with on your own time. A true breeding tall plant is crossed with a true breeding short plant. Tall is dominant. What would be the genotypes and phenotypes of the F1 offspring? And what are the genotypes and phenotypes of the F2s? So we won't go through that now, but this is for something for you to think about when you study. Now, test cross. you need to know what a test cross is and what you use it for. How can we determine unknown genotypes? Well, suppose you have a plant with purple flowers, and you know that purple is dominant. But you also know that it can be purple if it's big P, or big P, little p. You don't know which. However, you have another plant with white flowers, and you know what its genotype is. It's got to be little p. So if you want to know what the genotype of the purple flowered plant is, cross it to the white plant. In which case, if the purple plant is big P, in other words, homozygous dominant, you're going to get an F1 that is all purple, because they'll all be heterozygous at the P locus. If, on the other hand, the original purple plant was a heterozygote, big P, little p, then half of the F1 will be big P, purple, and half will be little p, white. So with one cross, you can determine the genotype, which is unknown and is masked by the phenotype. So that's what a test cross is, and that's what you would use it for, to determine the genotype of an unknown. Just a statement of the process. An individual of unknown genotype is crossed with a homozygous recessive. What happens if you're trying to follow more than one characteristic at the same time? Well, in this case, it's called a dihybrid cross, where plants with smooth yellow seeds are crossed with wrinkled green seeds. The F1 generation, they're all smooth and yellow, which tells you that those two characteristics are dominant. And the F2, the segregation is 9:3 to 3:1. And you can see why that is the case if you draw what's known as a Punnett square. Dihybrid cross, and here you can see why you get 9 of the smooth yellow, and the other ratios as well, the 3 and the 1, homozygous recessive at both loci.
So we get 2 outcomes from 1P generation.
2 outcomes.
Yeah.
Are you talking about a dihybrid cross?
Yes.
There's just one outcome, which is the ratio of phenotypes and genotypes that you see here.
Are you referring to the F1 generation?
Yes.
I'm sorry. Okay. Yeah, there is only one outcome for the F1 generation. And I don't know if you'll have time to work out maybe a problem or two before we're done, Professor, but if people would like practice working out Punnett squares, which I strongly recommend you get some practice working these things, please remember to show up to workshops, because I can spend time going over these. They're not terribly hard, but they can be a little confusing at the get-go. But once you do 1 or 2 of each type, monohybrid or dihybrid, they become far easier. And so these are things that we can certainly reinforce in workshop.
Yeah, I'd encourage you to do that. And Scott, your workshop starts at 2 today, is that right?
Yes, sir. 15 minutes after class.
Yeah, okay. Okay, to review Mendel's laws of inheritance. Segregation. So chromosome pairs and their alleles are separated into individual gametes to transmit genetic information to offspring. This happens when chromatids, remember each chromosome is a pair of chromatids, separate and each goes into a haploid sex cells, and this occurs at telophase 2 of meiosis 2. So going back to when we talked about meiosis 1 and meiosis 2, the segregation occurs in telophase of meiosis 2. Dominance, a dominant allele completely masks the effects of a recessive allele. Dominant allele has the same phenotype in both heterozygotes and homozygotes. That's dominance, another law that Mendel first established. And finally, independent assortment. The alleles on different chromosomes are randomly distributed to individual gametes, and this happens at metaphase 1 of meiosis 1. So again, go back and review what happens in the various cell divisions in meiosis. Independent assortment occurs at metaphase 1 of meiosis 1. So it might be instructive to go back and actually look at those illustrations of cell divisions in meiosis. So the last slide I have today is a summary of what we've talked about. First of all, the keys to Mendel's success, inbreeding, and analysis of the F2 generation. Those were critical, and apparently no one prior to him ever did that. And then I won't go through all these terms, but they're terms that have come up in the discussion today. And you need to know the definitions of each of these. Now, I have put in Canvas this slide set, as well as a worksheet in which I ask a number of questions, some of which include the use of these terms. So please look in Canvas. You can review the slides, and I will put a recording. I'll send you an announcement with the URL for the recording of this class. And at the next class period, we'll discuss the worksheet and the answers to the questions. So that's what I have for today for you. Any questions or comments about Mendelian inheritance or ligers?
Professor, did we go over polygenic inheritance?
Polygenic. Okay. Can you see this without me going to slideshow?
Yes.
Okay. So this is what you're talking about, right? What happens if you're trying to follow more than one characteristic? A dihybrid cross. So the two characteristics are smooth and yellow in terms of seeds. So smooth seed coat, physical characteristic, and yellow color, as opposed to a wrinkled seed coat that's got a green color. Each of these is controlled by a separate gene. So if you cross them in the F1 generation, all of the loci are going to be heterozygous. And so only one phenotype will show up. and that is smooth seed coats, yellow color. When you go to the F2, you see segregation in this ratio. And you see this best when plotted out. If you look at the gametes, so big
And
S,
how do we know it says that heterozygous?
how do we know?
That it stays heterozygous?
In the F1,
Yes.
how do we know that it is heterozygous in the F1?
Yes.
By both the genotype and phenotype. The phenotype, okay, so green and wrinkled disappear in the F1. So that tells you that smooth and yellow are dominant. It's all the,
I'm sorry, if I may throw something in. This is not something you would run one cross on and go, oh, that's just the way it happened. Like Mendel, you'd have to run hundreds or thousands of these in order to get a statistically significant result. So this is simplified. And again, if you did a Punnett square for the F1 generation, you would see that just by genetics, there is no way to come up with anything other than these heterozygous combinations. But your question was on polygenetic inheritance. And I'm thinking this course is only really going to concern itself with traits of alleles or traits that are controlled by single alleles. But what you're discussing is something more like where multiple genes affect a phenotype. And that's what polygenetic inheritance is. And it's really kind of beyond the scope of this course. Interesting topic, but I mean, I was a junior in my second semester in college in a genetics class before they started talking much about something like this, Jazzy. So I can't give you a really, really great answer on it. If you come to workshop, I can maybe discuss it a little bit with you, but generally in here, we're either talking about generally in here, we're only talking about traits that are controlled by a single gene. And so Mendel was lucky that the things he was looking at were actually single gene controls or single gene controlled. But because we're looking at two different traits does not mean we're looking at polygenetic inheritance. We're simply looking at two different traits that are each controlled by a single allele. For instance, color is controlled by one gene, and wrinkled or smooth is controlled by another gene. But putting those two together does not make it polygenetic inheritance. Does that kind of clarify maybe what you were asking?
Yes.
Okay. And if you have more questions about that, because I know we're starting to run out of time and I want to give this back to Dr. Yoder, you can stop by the workshop and I can try and explain it to you, but I will admit to having limited knowledge on this. And I can mainly just provide you some generic examples. Okay.
Okay.
All right.
yeah, so for more, I guess Scott call it practice with this, you can do that in the workshop. Next time on Wednesday, what we're going to talk about is molecular genetics. Okay, anything else about this class? Well, if not, you have the slides. You will have the recording, and you have a worksheet in Canvas that you can work on. We'll discuss the worksheet on Wednesday.
The worksheet is due Wednesday.
It's not due. It's for your purposes only. It's to help you study.
I also have some practice questions for these things because it's almost a guarantee you're going to have to work out at least a monohybrid, maybe a dihybrid cross, depending on how diabolical Dr. Yoder's feeling on the exam. They'll come back to you, and as I say, they're pretty easy once you get the knack for it, but sometimes it takes 2 or 3 iterations. So I have some practice problems. If you stop by the workshops this week or before the exam, I realize workshop doesn't fit with everybody's schedule, but I do have practices that we can go over, and we will unconfuse this if anybody has any lingering doubts.
Yeah, Scott is right. It will show up on an exam at some point.
And I